the theory and some interesting popular science non-fiction books that might be interesting for exploring ideas beyond class. So I have posted that in the comments of last yesterday's lab lecture. So please feel free to go there and look at it. If I have missed out something that I promised yesterday, do let me know and I can share these links with you. As I think the physical classes might begin after minor one. I'm not sure about that, but there was a government advisory today, a general public government advisory. So yeah, there is also physical copies of some of these books, and yeah, I'll be happy for some of you to borrow it from me. All right, so my screen is visible. I hope.
Yes, sir.
Yeah. So today, I'm going to introduce you to next set of algorithms, which are very important in biology, and I think a lot of you have already been enthused about probably how is it that we can sequence a genome of an unknown species, and this is akin to solving a problem of a puzzle with million pieces. And here I will be introducing you to a class of algorithms called graph algorithms. So this is the outline. So first I'm going to do an abstraction of the problem through what's called the exploding newspapers problem and how does it relate to genome sequencing. Then I'm going to formalize it like I have done before into a string reconstruction problem. And then we are going to solve the string reconstruction problem by two different methods, each of whom have their advantages and disadvantages. And then specifically, we are going to focus on the method of using Eulerian paths to solve the problem. And here I'm going to introduce you to another set of formalisms, which you can use to describe data and assemble genomes. And then at the end, I'm going to talk about some of the challenges that one would face in genome sequencing in terms of the real applications. All right, so if you had a stack of newspapers kept like this, let's say this newspaper represents the New York Times newspaper on June 27th, 2000, and somebody goes, puts a pile of dynamite under these papers and hypothetically blows them up. All right. Now, your task is to essentially take these pieces of paper, right, newspapers, and put it back together, put these pieces back together and figure out what did the paper on June 27th, the year 2000, the New York Times said. All right. So why am I talking about this problem? Because some of you might have guessed the newspaper here is abstraction for a complete DNA molecule or the genome. Multiple copies of these newspapers represent multiple cells from which this genome might be extracted. And the fact that you are blowing up these newspapers, which gives you random pieces of paper, is similar to the act of DNA fragmentation of these multiple copies of genomes. Okay, so the idea that you have to take these pieces and overlap them is what's called the overlap puzzle problem. Now this again, what does it have to do with biology? When you talk about a DNA molecule, it's double stranded, right? So essentially there are certain things, for every A there's a T on the other strand and for every G there's a C on the other strand. So this essentially means that the nucleotide is actually one half of this double stranded DNA that you would be having. So that essentially means that if you have uh, the sequence of one strand of DNA, you get the sequence of the other strand of DNA, the complementary strand for free. All right, so if we talk about the human genome, genome sequencing essentially means reading the genome, okay? So sequencing essentially is the term derived from the legacy of being able to sequencing in the context of reading letters of the genome. So now when we talk about the human genome, it is composed of 3 billion letters on a single strand. And if you consider both the strands, then it's single strand of single set of chromosomes. And if you consider both sets of chromosomes, you have 6 billion letters. And they basically look on the very, uh, on a page, these ATGCs just spread out. If you compare it with an amoeba, which is one of the longest known genomes, it has approximately 670 billion nucleotides, right? So almost 3 orders of magnitude more. Can somebody tell me what is the size of the largest genome and to which organism does it belong to? I asked this question earlier as well as a homework problem. Can you guess whether it's an animal, whether it's a bacteria, whether it's a fish, whether it's a tree, whether it's a plant? So the largest genome is from a plant. It's called the water lily, Japanese water lily. So far, no. Okay, but the key point is that the DNA is microscopic. So the question is, how do we actually read something that we cannot see? And in order to read it, why do we would want to read it? Because as you see, this is the figure of how different species of birds, specifically one species of birds on an island where these birds were isolated. This is uh, drawing from Charles Darwin's book, the famous drawing which captures how Darwin's finches or specific species of birds, finches, evolved on an island. And this is how, after you have sequenced the genomes of lots of different organisms, this is how they look like and they relate to each other. So you would like to sequence the genome to basically unlock the genetic identity and where do you place an organism in the context of all the other living organisms in the world. The other thing that you can also achieve by sequencing the genome is to actually save lives of people who suffer from rare disorders. So Nicholas Volker, this child here, he suffered from a rare disorder and his complete genome was sequenced in 2011. And because of sequencing of this complete genome, they were able to identify that a specific gene was mutated in Nicholas, because of which he was not able to make a specific protein, for which he was given that protein replacement therapy, and his life dramatically improved. And doctors did not know what was happening to him, uh, and they said that he's most likely going to die, but they were able to save his life. So here is another one of those homework problems that I often give to you. And I'm again telling you, these are things that I will definitely ask some of these things in the quiz that's going to happen next week during the lab hours on Wednesday, the first half. Please go and read up about Nicholas Volker and what gene was responsible for his condition and what does that particular gene do. All right, so the history of genome sequencing we've already talked about in the last module. It was developed by Walter Gilbert and Frederick Sanger, and then later on, due to lots of technological innovations, it was made into a high throughput method. The first genome that they sequenced was the phage genome, bacteriophage genome, which was about 5,000 nucleotides big. They shared a Nobel Prize in 1980, but their approach was quite expensive because it cost about $1 per nucleotide. Using that same cost projection in 1990, Francis Collin, he actually visited IIT last year, and was given the Human Genome Project to lead the Genome Project. And because we have 3 billion bases, he was given $3 billion dollars to work on this project. And similarly, in 1997, another one of genomics researchers, Craig Venter, opened a company called Celera Genomics with the same goal. And by now, I hope all of you can distinguish between the methods that they were using. So the Human Genome Project was using the hierarchical clustering, hierarchical sequencing method, and Craig Venter was using shotgun sequencing method. In 2000, the first draft genome was published with both of the people there. And since then, many genomes have been started. Many more genomes have been sequenced. So the cow genome was sequenced more recently in 2009. Then you had the horse, the mouse, the macaque. So macaque is basically the monkeys that live very close to where human settlements are. Chimpanzees, dogs, rats, mice. So here, mouse, rat, dog, chimpanzee, macaque, these are all organisms on which a lot of pharmaceutical testing happens, and cows are important for animal husbandry and agriculture. So, but many more were sequenced during this using the Sanger approach, high throughput Sanger approach, in cahoots with a little bit of 454 sequencing. But this definitely did not scale for many more genomes. So therefore, there were new technologies that were developed that we discussed in the last class. Illumina was predominantly the most popular technology that was highly adopted. And because of this, from early 2001s, where it basically costed uh, approximately $1 per base, this cost $1 per nucleotide. The sequencing cost by 2019 has gone down to 1, 2, 3, 4, 5, 6 orders of magnitude low. And in 2022, now the cost has gone down to one more order of magnitude. And as you can see, this cost is the speed with which this is going down is much more faster than even the Moore's law, which determines how many transistors you can pack in a chip physically. So this has led to many big sequencing projects, as I have introduced earlier. You have the Genome 10K project, where the idea is to sequence 10,000 genomes of different vertebrates. There is the I5K project. So I stands for insects. So they want to sequence 5,000 different kinds of insects. There is the B10K project where they want to sequence 10,000 different kinds of birds. Birds are very interesting because they have very interesting courtship behavior, and they are quite smart animals, and they are known to be on Earth for a very long time. So therefore, there's a lot of interest in bird brains and how bird social behavior exists. And there are also a lot of projects around the world on a governmental scale. So when I say BGI, that essentially means there is definitely a project going on in China where they have probably sequenced more than 10 million humans already. Um, then there's what a project called 100K project. So basically, residents of United Kingdom from 2012 to 2018, they have already sequenced about 1 lakh genomes. And then there is the Genome India project, where the idea is to sequence 10,000 Indian genomes. And this project is ongoing. It will be finished in 2022 this year in August, and the results will be announced on Republic Day, not on Republic Day, on Independence Day, sorry. So just to give you the overview of genome sequencing, I'll just repeat what I have been telling you earlier. So you have multiple identical copies of a genome. They are shattered, just like the newspaper, into small pieces. These pieces are put in a sequencing machine, and after getting read in the sequencing machine, these pieces are called reads. You sequence these reads in the lab, and then you overlap them. And once you have overlapped them computationally, then you can reconstruct the original sequence. All right. So what does the genome sequencing remind you of? It basically reminds you of the overlap puzzle, which where I showed you initially that the newspaper is blown away into bits and you have to reconstruct the newspaper pieces. Okay. So now I'll go into the formal definition of the problem. But before I go into the formal definition of the problem, there are certain complications with genome assembly. So the first complication is DNA is double-stranded, and it may consist of multiple independent chromosomes when you're talking about organisms like humans. The second is that the reads might have imperfect coverage of the underlying genome, which essentially means that not every portion of the genome might be covered by all the reads in the same manner. The third thing is that sequencing machines are error prone. That means that the sequence that you are getting of the read might not always be exactly a true representation of that fragment. There are errors that happen in the process because it's a chemical process at the end of the day. Okay, so to make our lives easy, we make some assumptions and we solve the case where these complications are not there. So what are the assumptions that we make to understand and develop a algorithm for genome assembly? First assumption is that the DNA is single stranded and it consists of a single chromosome. So that essentially means single stranded assumption is quite fair to imagine because if you get one strand, you'll get the other strand because of complementarity, and single chromosome also makes sense because if you are able to solve for a single chromosome, then essentially the same ideas can be applied or tweaked to solve the problem of multiple chromosomes. The other big assumption that we have is that reads have perfect coverage of the underlying genome. That essentially means that every substring of length k is generated as a read. All right. Essentially meaning that every substring from the genome is going to be represented. There is no region of the genome from which there are k-mers missing, or there is no representation of k-mers from those regions. And obviously the third one is the sequencing machines are error free, which is almost the truth because these errors happen 1 in a billion depending upon the technology that you're working in. And anyways, most of these errors can be learned by the machines, and newer AI algorithms, artificial intelligence algorithms are essentially making the errors even lesser. All right, so with these assumptions, we move forward to developing a formalism for genome assembly problem. Okay, so let's try. So the first thing is the genome assembly problem is we have to reconstruct a genome from the reads. So everybody knows what reads are. Let me just stop here and ask you that question. Can I see a show of hands from all of you who can now confirm what a read is? Anybody who's still confused about what a read is? So I see 5 hands, 6. Okay, so probably six of you are listening, which is usually the percentage that I expect. Okay, 7. Okay. Does anybody not understand what a read is at this point of time? What a read is, or what read length is? Please raise your hands if you don't understand what a read is, or what read length is, or what coverage is. Only raise your hand if you don't understand these words. Ratnesh, you don't understand these words?
No, sir. I understand. I understand.
Then you can please keep your hand down then. Okay. So, so the input to this problem or to this algorithm would be a collection of strings in the form of reads, and the output would be a string, which is a genome, which is reconstructed from the reads. So typically the reads are going to be shorter in length than the genome that they are representing. So it's not really a clear computational problem because we haven't defined what does reconstructed mean, right? We don't have a clear definition of the word reconstruct. Like it's just putting it together. When do you stop to put things together, right? So we have to come up with another way to define the problem. So this is very important. Like whenever I'm starting any of these algorithm development in every module, you have to understand that even for you, if you are starting with any kind of algorithm or trying to solve any problem, it's not a bad idea to first define the problem in simple words and then trying to then understand whether you can explain the computer, or like in computational language, can you very precisely explain what the problem is, right? So in this case, the problem that we have is we have to explain the computer what do you mean by reconstruction. So in order to do that, we have to come with a mathematical formulation of what do we mean by a reconstruction of a genome from reads. So let us say that k-mer composition of a text. So I'm introducing this new term. The k-mer composition is denoted by this function composition with a subscript k of text is basically a collection of all the k-mer substrings of the text. This includes repeats. Okay, so let's say this is the composition. So the k-mer composition of TAT, GGGG, TGC is essentially this. It's AT, GGGG, GGT, right? Like this. Now here you'll notice that I have written the composition in a lexicographic fashion, right? Because I don't know when I will be getting the reads, in what order I will be getting the reads, right? These are just random fragments. So it's safe to assume that we can organize them. One thing that we do know about the reads is that they can be organized in the form of some lexicographical order. That's how they will appear in a dictionary. So A comes first, T comes later. So you essentially organize the reads like that. All right. So now I want everybody to please take a pen and paper and be ready for some class problems that I'm going to give you. I want some participation here now. Okay. All right, so the string reconstruction problem essentially is to reconstruct a string from its k-mer composition. All right, how do we define it? You are given a collection of patterns of k-mers and you have to come up with a string text whose k-mer composition is equal to patterns. All right, so you have collection of patterns of k-mers and the output, if the text that you have is equal to the k-mer composition, right? The k-mer composition of the text is equal to the patterns, right? If you take a text, its composition is equal to the k-mers that you have, the read that you have got, and that's like a complete LHS equal to RHS kind of a formalism where you can write a clear algorithm for that. But here again, the problem is that we don't really know uh, if what happens if the patterns are non-overlapping, right? If there would ever be a string that exists with certain weird patterns that are there inside the gamer set. Okay, so now we have to define the problem even more precisely. And for this, I will again, please, that's why I'm saying, please take your pen and paper and maybe work these problems that I'm doing along with me in the class. So let us reconstruct the string corresponding to the following three-word composition. So can somebody tell me what is the string that is represented by the following three-word composition? So let's try. So this is how it looks like. So you have TAA, you have AAT here. So you go put it like this. Then you have ATG is here. So this overlaps this. Then you have to look for a TG. So TG is here. So this is a TGT, and then you have a GTD, right? So this is the three-word composition. So this text, three-word composition of this text is equivalent to these following k-mers, right? Is that clear?
Yes, sir.
Yes, sir.
So this is a greedy algorithm, right? As we have seen earlier. So in greedy algorithms, the idea is to be greedy, is to try all possibilities, the simplest possibilities, and you essentially answer what you end up with. All right. So basically, in this case, the greedy algorithm says for each k-mer, look for the k-mer with maximum overlaps in each direction, and then put them together. This is how typically you will be solving any puzzle even, right? So then genome assembly seems quite trivial and we can just go home. Okay. So now I'm giving you an example and I want everybody in the class, whoever is listening, to please solve this problem by yourself. Okay. Please apply the same funda that I gave you. I'm giving you 3 to 5 minutes to come up with a quick, like tell me which string will have the k-mer composition equal to this, what I've given here. And if you already see that there could be certain interesting discrepancies coming up. So again, if you can look at this, these are in lexicographic order, right? So you have a AAT, ATG, 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 CAT, CCA, then you have Gs, then you have Ts, right? So it's all sorted by lexicographic order.
So I don't think like using like this method we can justify which game or which text is unique.
So you can. So you were not able to?
So the thing is like from AAT you can go to ATG, but from CAT also you can go to ATG, and from GAT also you can go to ATG. Then from ATG you can go to TGC or TGG or TGT.
So there are
And
multiple
one.
possibilities here, right? So let's
Yes,
start
sir.
here. Let's take one of these things. So let's start with TAA like we did earlier. We have TAA and AT, right? We did that earlier as well. Now we
Sir,
have an AT. Yeah.
sir, is the length defined, the total length defined that we have to construct by using all these schemas?
Yeah. Because you are
Because
sequencing a genome, right? There is only reads only fragments from that genome. So that is that assumption. So you have perfect coverage. So whatever k-mers that you are getting have to be part of that genome.
no, sir, I am asking because it will depend vary upon the length that how much big string we have to create using these small parts, these schemas.
So that's what I'm saying. No, so you have to use all the k-mers. So if you have, let's say 10 k-mers of length 3, then the length of the genome that you will have to construct finally is,
So
can you tell
12.
me?
12.
Are
Maybe
you sure?
12 may not will because there is a repetitive ATG, ATG, ATG. Then how can we
You have
say
to use
that
all of
12?
them.
Yeah, but there can be repetitive sequences, right?
So we can
There is no
just
repetitive
like
sequences here.
you have n minus k plus 1 kmer, so you
N
can
minus
just
k
like
plus 1 into
transpose it.
3 plus 2.
Yes.
No, not 3 plus 2. Sorry. It's n minus. So yeah, it's the n minus 1 k plus 1. So you have basically 10 k-mers. Yeah, so yeah,
Yes,
overlap
sir.
plus 3.
So
So 12 is the
like
length.
yeah.
Okay, so let's go here. So let's take one of the ATGs here. Okay. Now, which, so this is what the problem is, right? So which of these other k-mers should we choose, right? Each of the k-mers might lead to a different path. So choose this one, TGC, GCC, then CAA, CAT, ATG. We are using this. So the ones which we use at any point of time are we've used are gray. The remaining ones are black, and the ones that we are currently trying to overlap are in red. So now let's take the lexicographic order. We take again the top one, TGG. Now we again take the top one, GAA. Now we have again only one choice, GAT. Then we have ATG. So we have used all ATGs. Then you have GTG. So what happens to GGG, right? So at some point of time, we had taken a wrong step because of which this particular gamer was left out, right?
Yes,
So
they placed at one place.
yeah. So
Uh,
I think Vishantan already said a few issues with this algorithm. So can we repeat? So one clear issue is what is the biggest issue here?
So when the last base is different, like you can have multiple possibilities.
So because you're doing a partial overlap every time, whenever you have multiple possibilities, you don't have a clear idea of which one to choose, because until you choose and go down that path, you are not sure if your choice is correct. Okay, is that clear to everyone? Again, I'm
yes, sir.
going
Yes, sir.
basic, and then I'll take it into more tougher region. All right, so the greedy algorithm essentially fails because of these kinds of repeats or these kinds of repeated overlaps, right? So in this case, the repeat will be the fact that you have TG, that Kmer TG coming up multiple times. So again, this exercise, something that I've asked you to do earlier, can you tell me how many 300 mer DNA repeats can you expect randomly in a DNA string of length 3 billion? Again, please take 2 minutes and It would be great if somebody could tell me the answer. What do you think are the odds of a repeat of length 300 in a random DNA sequence of length 3 billion? Anybody wants to give me a formula for this?
So first from like, since we have 300 merge, so we choose any two out of the 300. I'm sorry, since we have uh, like 3 billion minus 300 plus one possible 300 merge, we choose any two out of the like those quantities. So that would be like, if we have n number of 300 merge, so it would be NC2 into 1 by 4 to the power 300 to the power 2 into 1 minus 1 by 4 to the power 300 to the power n minus 2.
Everybody agrees?
Sir, I did not understood.
Okay. So yeah, so this is
But this is only if you have like a double repetition. Like if you want
exactly.
multiple repetitions, we can do like 1 minus no repetition.
Yeah. So this is not so easy. Again, I'm saying, telling you, this might be one of the questions that might come in the minors. So please figure out how to answer this. So actually repeats are very common in genomes, as we have already talked about. One of such repeats, 300 nucleotide long repeats, is the ALU repeats, which occur over 1 million times with very few minor changes in almost every human genome. So dealing with repeats is a problem, and we have to come up with a way to deal with them. And what the remaining part of the module is going to give you some ideas on how to deal with them. So an interlude here. I hope everybody understands how sequencing happens in the Illumina machine, at least,
Yes, sir.
Yes, sir.
how it happens in the BGI machine with the DNA nanoballs,
Yes,
Yes,
sir.
sir.
how does it happen in the 454 machine in the pyro sequencing, and how does it work in the nanopore?
Yes, sir.
Okay. All right. So now let me define the first formalism of solving the string reconstruction problem as a Hamiltonian path problem. Okay. So when you have uh, such kinds of repeated regions, you do have solutions, as I show you here by just not following the lexicographic order as we did earlier for looking at overlaps, read overlaps. We can essentially do like with a few hidden trials, we can come up with the right sort of overlaps between k-mers to reconstruct the original string so that no k-mer is left behind. And here, in order to do so, we essentially have to view um, this entire overlaps as what's called an overlap path graph. So the fact that two k-mers share a two-mer overlap can be used to define this graph. So for example, this graph essentially can go from left to right, where the two nodes or two k-mers can be represented as nodes. Each node is a k-mer, and the arrow above them represents the overlap. So for example, you have TAA and AT. So the arrow essentially is representing the overlap AA. All right. So this is what's called a genome path. Assign each read to a node, connect the adjacent nodes, adjacent reads with edges. So can you see the genome in this? If I give you a graph like this, can you tell me the genome quickly?
Yes, sir.
Yes, sir.
Is there a fast way of doing it?
So we just have to take the first element and add the last letter of each
Exactly,
subsequent element.
right? So it's TAA, TGCCAT, right? Like that. Okay, so now could you construct the genome path if you only knew the 3-mer composition? Is that possible?
Sir, what is genome path and three-more composition just like this above?
Yes.
Yes, then exactly we can.
Yes, sir.
Oh, yes, sir.
So please wake up all the people. I mean, these are simple things that I'm telling you, but please be, it's because this is some mathematical formulation, okay? So you know the
Yes,
genome path. Could you construct the genome path only with a 3-mer composition? A 3-mer composition means that you are only given these 3-mers, right?
Yes, sir. We
So
need to know the order.
the 3-mer composition essentially, so let us say the 3-mer composition of the word genome is GENENONOMOME. So if I'm only giving you the 3-mer composition of genome, I will just be giving you the 3-mers of the word genome, right? And we have clearly shown that if you are only given k-mers, because of the process problem of repeats, you cannot always complete, right? Create paths like this. Otherwise, you have a solution to the genome reconstruction problem. We need to know the order of k-mers. Okay, so now let's define how we do ordering of k-mers. So a prefix of a k-mer is k minus 1 letters in the k-mer. The suffix of the k-mer is k minus 1, last k minus 1 letters of the k-mer. Okay, so the prefix of TAA is TA and suffix is AA. Clear to everyone?
yes,
Yes,
sir.
sir.
Okay, so an overlap graph is formed when you have a node from each read, okay, in the set of patterns. So patterns here represent sum of all the reads, and then you connect an x to a y, you connect two nodes x and y. If the suffix of x, if the suffix of x, AA, is the prefix of y, AA here, right? If these are same, then we connect the nodes. Okay, so let us see. So you have a graph like this. Now you also connect, so you have a connection like this, but you also connect AAT with ATG. So you have to connect this as well, right? You also have to connect it with the other ATG, right? Like this. When you start to connect, the graph becomes kind of complicated. Okay, is this clear?
Yes, sir.
Yes, sir.
So
Yes,
given
sir.
a set of k-mers, if you have to construct the overlap graph, you have to give it this particular formalism that you connect two nodes if they have the prefix of the node below and the suffix of the node above is the same, then you connect the graphs, the points. Okay, so now when I say I have to find the path or the genome here, right? Can you now think about a way, a mathematical way, given that you have constructed a graph to reconstruct the genome? Let us say I've given you a graph like this, where all the possible nodes are drawn. Can you still see a genome path here?
No, sir, not exactly.
But what will be the properties of a genome path if it exists? If such a path exists? Can you give me, can you think about a definition?
So
If
it
a
only
genome,
goes
I
through
say that
each
if a
element
genome path
once.
exists in this graph, what can you define what that path will look like?
So it would only go through each element once, and like it covers all the elements.
It goes through each element once and it covers
All the elements.
all the elements. So element here is nodes.
Yes, sir.
So if we have a path existing, a good genome path would be such a path where you cover each of the nodes once. Does that make sense?
Yes, sir. So it's like a bijective function basically.
Right? But here your life is quite easy, right? Because we have constructed this path from the original path that we know that the path in between is the correct path, right? Because we know what genome it comes from. Yes, everybody is with me.
Yes, sir.
Yes, sir.
In reality, we will not get it in this fashion, no? In reality, we don't know when we are sequencing, whether TA will come first or AAT will come first. In reality, we'll be given jumbled all these ATGCs, all these reads. And typically, the graph that you will be creating would actually look something like this. This is a lexicographically ordered graph. You have a AAT, you have a ATG, 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 CAT, CAA, GAT. If I arrange it lexicographically, the genome that was apparent earlier sort of vanishes, right?
Yes, sir.
It would be tough to find the genome path in such a graph. So basically, here I introduce you to the concept of the Hamiltonian path in an overlap graph like this, which basically says that it's a path through a graph that touches each node exactly once. So basically, if you are able to touch each node once in a overlap graph, then you have essentially found a path that represents the entire genome. Is this clear?
Yes, sir.
Yes, sir.
Show of hands, please. This is very important. This is simple concept, but I want to see how many people are clear with it. Okay, good. So this is exactly how one solution looks like, the solution that we had. And it's, as you can see, it's not trivial to find this solution. All right, but there can be another solution also, right? So the graph organizes our reads, but we don't have an algorithm for finding a Hamiltonian path. Okay, so we have to basically come up one part of the graph, one part of the problem is sort of solved, that if we have k-mers, we have to organize these k-mers in the form of a graph with the definition of suffix and prefix of two nodes being the same. You connect the graph, but now we have to find a Hamiltonian path in the graph. So we have to find the algorithm to find a Hamiltonian path. Okay, so what does the overlap graph look like if there are many repeats? Can you find some characteristics of this graph where using which you can actually figure out that there are many, if this graph has repeats or not?
Sir, what does it mean by repeat here, sir? Like,
Repeats means it has repeated elements.
So you
like,
could have one node connected to multiple nodes if there are repeats.
So everybody agrees with this answer? If one node
uh,
is connected to many nodes, then there are repeats.
Necessarily.
sir, how can?
Not necessarily.
So let us see.
Yeah, sir, not
Let us see where
necessarily.
the repeats are. Okay. So ATG is a clear repeat, right? So you see many
Yes, sir.
lines.
Yes, sir, yes.
Many lines
Yeah.
are coming out of ATG and many lines are going into ATG, right? So if you have nodes, so I'll define another concept here, the in degree of a node and the out degree of a node. So the number of arrows which are going inside the node is called the in degree, and the number of arrows which is going outside the node is called out degree. All right. So if the in degree and out degree of the nodes is more than 1, then they are most likely repeats in the graph. So verify the statement, okay?
Yes, sir.
All right. So
Yes.
the genome assembly problem essentially, as I defined earlier, was essentially the genome reconstruction problem. And was a genome reconstruction problem. And the reason why I actually talked about the newspaper on June 27th, 2000, New York Times, because this is what the New York Times was saying when you overlapped. So you have two pieces, and when these two pieces came together, right? So instead of using overlapping fragments from different so when you are trying to reconstruct, you use overlapping fragments from different genomes essentially, because the same genome, when you're fragmenting, the fragments by the nature of the process of fragmenting are non-overlapping, right? Because these fragments are coming from different genomes, so therefore it fits like a jigsaw puzzle. And the reason why I actually showed you was because this is when the Human Genome Project was announced, and this is how the front page looked like. Okay, so the genome assembly is essentially equal to the exploding newspaper puzzle, and we essentially, this is how it looks like. No technology is currently there to read the nucleotides of a genome from beginning to the end, just like we read a book, but technologies are coming up like nanopore. The best we can do is to sequence much shorter DNA fragments, uh, also known as reads. And this allows us also to do a lot of simultaneous sequencing, because we can, as I showed you, when you are doing sequencing, you are, because of the fact that you are fragmenting, you can actually simultaneously sequence a lot, because you can simultaneously sequence.
So I had a
uh,
question here.
yeah.
So like if like I guess we can have an overlap path which has multiple overlap graph which has multiple Hamiltonian paths.
Yeah,
So like what do people do in that situation?
I'll talk about it.
Okay, fine.
Okay, so a
Sir,
genome assembly is
uh,
basically a string reconstruction. You have more question?
Sir, I have a question. In case of we have gamers of k is equal to 3. We have like multiple parts, but when we have when we are sequencing human genome and we have fragments of big fragments, 100 kb, 100 base pair and 200 base pairs. In that case, if we had matching 20 base pairs are overlapping, then that kind of repeat will not be found in so much.
True, true. I agree.
Yeah,
So right now, Ratnesh,
it
I'm
will be
just
easy.
showing you
Yeah.
a simple example so that you can understand how these algorithms work. In
Yes,
reality,
sir.
the length of k-mers is much more. So k-mer lengths are usually 30, 37, 43, things like that.
Yes, sir.
Okay. So genome sequencing is essentially a, you have more question?
No, sir.
Okay. So I'm just repeating myself here as I end the class. So reads are generated by modern sequencers, often have the same length. Okay, so we can safely assume that the reads are all k-mers of the same value of k. All right, so can somebody tell me why is this the case? What part of the sequencing pipeline that I have talked to you about ensures that the reads are almost of the same length?
Sir, like in alumina, we have those short fragments where this
This is even before
reading.
Illumina.
Yeah. So, sir, like in shotgun sequencing, we don't think all the
This
fragments
is
will be of
before
same
even
size.
shotgun sequencing was coined.
Maybe, maybe of same size. What did you say?
This is even before shotgun sequencing.
Okay.
So what phenomena during the sequencing process, which even during Sanger sequencing people were utilizing, ensures that we have k-mers of the same length?
Sir, cloning.
No.
Sir, we use those magnetic beads to extract multiple time use those magnetic beads to extract the same length of the cameras.
So what was the step called? That step was called size selection,
Size selection, yes, sir.
right? You can also do that by cutting the gel at a specific size. In
Yes,
ideal
sir.
situation is basically in which the reads come from the same strand, they have no errors, they exhibit perfect coverage. That essentially means that every k-mer substring of the genome is generated. So when I say a k-mer composition K of a text, is basically this is how the k-mer composition will be written like, and typically you would represent the k-mer composition in the lexicographic order. So genome sequencing essentially is a string reconstruction problem. As you have an input, you have a string, you have integers, k, an integer k, right, and a collection of patterns of k-mers, and the output is a string text with the k-mer composition equal to the patterns if such a pattern exists. All right. I've already showed you this. I've already showed you how the Hamiltonian looks like. And now we basically have to figure out a way to come up with the Hamiltonian path if there a unique path exists, or for example, or do we even have to actually look for a unique path here? All right. So this is where I will stop and I will continue in the next class and I will introduce you to another abstraction, which is called the Euler path, which shows a representation of k-mers and another way of representing this overlap graph, which makes this which provides a different solution to the problem. So basically, finding the Hamiltonian path is not trivial, and there is no clear a single solution for finding the Hamiltonian path. So therefore, this is something which people haven't pursued, and I will talk about it in the next class. So I'll stop here. If there are any questions, I'll take those. Otherwise, you are free to leave for the next class.